So hello to everyone. I'm Dick Miles. I'm the board chair of the Foundation for Barnes Jewish Hospital. I'd like to welcome everyone to the Foundation for Barnes Jewish Hospital's annual Legacy Circle Luncheon. And just talking to some people in the last 15 minutes, it's been great. They've talked about their attachment to the hospitals, about the research that has affected their families, and I'm sure there are many, many other stories in this room that would be heartfelt and about the importance of Barnes Jewish Hospital and all the people that are associated with it. A few other board members and team members are here from the foundation. The board members have black and gold ribbons on their cards, so if you get a chance to talk to them, please do so. But today is really about you. This afternoon, we're delighted to honor you for your longstanding partnership and commitment to our hospital as part of the Legacy Circle. With your generous gift through your estate plan, you have made an investment in the future of Barnes Jewish Hospital and its partners. Your gift makes it possible for us to help more patients, nursing students, physicians, researchers, and our community. Together, we're improving healthcare for all our families and friends. At your table, you may note that we don't have traditional table numbers, but we have dates. These are important dates in the history of the hospital. And also, there is a new book that has been published on the history of Barnes Jewish Hospital, and if you don't have one and you would like one, just let us know. For example, in table 1892 is the year that Robert Barnes left a bequest of $850,000 to build a modern general hospital in St. Louis. Now, because of this gift and similar gifts to create Jewish Hospital, we now benefit from Barnes Jewish Hospital, one of the top hospitals in the country, if not in the world. Years from now, a group could be sitting at a luncheon like this, talking about the amazing impact that your bequest has had on healthcare. The bottom line is you're an essential part of everything we serve, now and in the future, and benefits from world-class, exceptional, compassionate care are the results. We're so grateful for each of you and what you do. We have a lot to share with you, but now let's take some time to get to know each other better over lunch. After lunch, we will hear from Dr. Ryan Fields, Associate Professor of Surgery and Surgical Oncology at the Washington University School of Medicine. So as we look forward to Dr. Fields' presentation, please enjoy your lunch, and thank you again for being with us today.
Good afternoon, everyone. Hey. For those of you who don't know me, I'm Bob Cannon, and I'm the president of Barnes Jewish Hospital. And we have some remarks here, and we'll be introducing our speaker in a second. But I wanted to pick up a little bit on something Dick was commenting on, which is each of the tables have a particular date and association with them. And the table we're fortunate to sit at is the 1892 table. And so the connection here is that last Friday, the chairman of the Barnes Jewish Board, Jim Weddle, and I have an annual event we do to honor Robert Barnes, the founder of Barnes Hospital. And when Robert Barnes died in 1892, so he is buried with his wife and children at Bellefontaine Cemetery. And it's really just a wonderful, serene setting and kind of family set of graves. And in his will, in Robert Barnes' will, he empowered 3 trustees, and these are names you'll recognize, Samuel Couples, Richard Scruggs, and Samuel Kennard, to use the money from his estate to build a hospital. And he had one thing in his will that was completely non-negotiable, which was he wanted a hospital to be erected as a hospital for sick and injured persons without distinction of creed. And that was an important founding principle for Robert Barnes. He went on to say that the trustees of the hospital shall not be paid any salary, reward, or remuneration of any kind, and that he had one modest request, which was that future generations maintain his grave at Bellefontaine Cemetery, quote, in the best repair and condition forever. So that's part of both the bequeath and the responsibility that we've taken on as his successors at Barnes Jewish Hospital. And so it was just happened to be an event that happened last Friday. You may see some pictures of it on our website and others, but it's really both a solemn and kind of inspirational moment to remember each year the generosity of Robert Barnes and the other founders of Barnes Jewish Hospital. So today I really get a treat. Dr. Ryan Fields is here. For those of you who haven't met Dr. Field, he really is a renowned researcher in the fight against cancer, and we're delighted that his wife Laura is here with us also today and able to join us. Dr. Fields specializes in pancreatic, liver, bile duct, stomach, and intestinal cancers, along with soft tissue carcinoma, sarcoma, melanoma, and other skin cancers. In 2017, Dr. Fields was named to the Best Doctors in America list, which recognizes the top 5% of all physicians. And in 2018, just this year, Dr. Fields was appointed co-leader of the Solid Tumor Therapeutics Program at the Siteman Cancer Center. Researchers in Solid Tumor Therapeutics Program study solid cancer sites to work toward improved cancer treatment and choices for our cancer patients. And Dr. Fields has a particular interest in treating cancer patients who are at a high risk for recurrence and in researching and using novel treatment choices to improve patient survival rates. Dr. Fields is just a dedicated scientist and researcher, a wonderful clinician, a great educator, really embodying, I think, all that is best in our Washington University colleagues. He's also a really great bicyclist who is an inspiration to me as a fellow cyclist. So since he came here as a resident in 2003, he's really been nationally renowned for patient care and research. And as he, I think, will share with you, is very grateful for the support that has come from the foundation. So Dr. Fields will tell you much more, and we're just thrilled to have him here today, and we'll invite him to the podium. So Ryan?
Thank you. We'll throw that up there. So as we're getting the slide set up, I'd like to reiterate one thing that Bob had said, and that's my deep gratitude to the foundation, both personally and as a representative of the Cancer Center and a lot of the other physician researchers and educators. I mean, I could not have done, meaning started a research program, without the support that the foundation provided initially and continues to provide. And it's just a source of unbelievable both financial and administrative and support for our program. So I was a little shocked when they asked me to speak, but I figured they asked me to talk after lunch because they didn't want anyone to pay attention. And I thought, if they really want me to talk for 3 hours, that'll be fine, and we'll just have an afternoon of it. But no, we'll talk for about 20 minutes. And the topic that I chose was cancer in 2018, current successes and new horizons. The reason I chose that topic is we've sort of gone through a huge revolution in the past 5 or 6 years in the way we think about and treat cancer that's directly relevant to all of us as researchers and physicians, but also to the audience. We always start, I have no disclosures or conflicts of interest to tell you about. A little bit of background that Bob had alluded to, but I'm a surgical oncologist, so I'm a surgeon. I spend about half my time seeing patients and in the operating room, and the other half on the research program. I grew up in Detroit, went to the University of Michigan for undergraduate, and then Duke for medical school, and then came to WashU in St. Louis and did my residency here in general surgery. Another interesting and serendipitous connection that's relevant today is our annual Resident Research Day, and that Research Day is named the Samuel Wells Resident Research Day. Dr. Wells was the chairman of surgery here for many years and was here when Barnes and Jewish Hospital united, and is really, I think, responsible for, within the Department of Surgery especially, making this a flagship academic institution of research and education prominence and excellence. And so we have a great day later this afternoon with our research residents showcasing a lot of their research, much of which was funded through the Barnes Foundation. So then I went away for 2 years to Sloan Kettering for my surgical oncology fellowship in New York, and then came back and joined the faculty. As was alluded to, I have an active interest in the biology of cancer metastasis, understanding how and why some cancers spread and some don't. And we talked about some of the solid tumors that I focus on clinically and in the lab. But this is also who I am. These are my two kids, Gabe, who's 9, and Sarah, who's 6. You can see somehow my wife and I only make redheaded kids. They're the apple of our eye, and we're fortunate to have enjoyed St. Louis. My wife Laura is born and raised here. We have a little lake house out at Innsbruck that people may be familiar with. We spend a lot of time out there. We go back, my son and I at least, and the family usually make a trip back to the University of Michigan to the big house. I'm sorry if there's any Ohio State people in the audience, you have to leave now. And my wife's parents are in St. Louis. They live up in the Creve Coeur area, and we spend a lot of time with them. It's wonderful. So what are we going to do today? Well, I'm going to kind of go through with a lot of analogies. I was happy to sort of look down the list of people who are here and not see a lot of MDs and PhDs after the name, and I think this is one of the more exciting talks that I get to give that's sort of geared towards an everyone audience. We'll sort of go through some common definitions and terminology, really talk about this cancer revolution that I alluded to, sort of talk about where things are now and where we sort of see things moving and how that's relevant both in the clinical side and in the research side. So let's start with a few definitions. If I have something that I want to define, I usually go straight to the definitive source of Google. So, if we really put cancer in and think about what is cancer, Google actually gives us a good definition. It's hard to read down here, but the disease caused by uncontrolled division of abnormal cells. And that's really the crux of it. If you really think back to what the problem is, every one of the cells in our body originated from one cell that originated back when we were conceived, and that one cell has all the information it needs on how to become any type of cell. And cancer is really a disease of those cells sort of working backwards. A cell that was supposed to be an intestinal cell gets confused due to some errors, and it starts dividing uncontrollably and thinking it's not supposed to be an intestinal cell anymore. And the problem is much like a virus that infects a person. The virus doesn't think a lot about the full effect on the host, so these cancer cells will grow uncontrollably. And what I think about a lot is the difference between a primary cancer and a metastatic cancer, and those definitions are important. When we say a primary cancer, we're talking about the original location where a tumor started. So the analogy I'll use is a colon tumor in the setting of colon cancer. It started in the colon, that's where the problem originated. Metastatic cancer, in contrast, is deposits or spread of cancer from that primary site to a distant location. So for example, a liver metastasis, so a tumor that's in the liver, but it started in the colon. So the tumor cells sort of shed from where it started and somehow got to the lung or the brain or the liver. And well, how does that happen? Well, if the tumor's growing, say, in the organ that's the colon, and there's blood vessels and lymphatic vessels, sort of the network of communication from the body, those cells can invade and get into the bloodstream and then travel through the bloodstream and end up in a distant organ. But that cell's got to figure out how to do that. So one thing that I like to point out to people is everybody in the world right now probably has a malignant cell in their body, a cell that made a mistake and through replication has a genetic error, but the body clears 99.9999% of those. It knows how to get rid of them because it's built into our programming. Cancer forms when one of those escapes somehow. It figures out how to trick the body and evades those systems. And if you think about the complexity of the cell or the human body, it's thousands of times more complex than any computer we have now. So there's so much going on in parallel at the same time that the body's able to really deal with most of this. It's those few escape ones that will turn into a cancer. So one of the facts that sort of comes of this primary tumor versus metastatic tumor is that the vast majority of patients who die of cancer, they die of the metastasis. You don't usually die of the primary colon cancer from the colon. You die of it spreading to the liver and causing liver failure, or spreading to the lungs and causing problems with breathing or pneumonias. It's very rare you die of that first tumor. And we understand how these primary tumors start pretty well. We understand what happens in a colon cell or what happens in a pancreas cell that turns it into cancer, and we understand that at a pretty deep level. What we don't understand is how metastasis happen. How does a cell start dividing uncontrollably and then invade into the bloodstream, survive in the bloodstream, figure out how to get out of the bloodstream, take up shop in a completely foreign organ like the lung or the liver and grow. And that's a process that people are really working hard on, our group included, to understand that. Well, why would that be important? Well, if we understood why that happened, we may be able to develop therapies that really target that problem or that pathway. And number 4, and this is sort of alluding to the next section, is until about 2010, most cancer treatments were very non-specific and non-targeted. The typical kind of chemotherapy, and we'll talk about that in a moment. So prior to 2010, we had chemotherapy, but chemotherapy is, it's poison. There's no sort of other way to say that. Chemotherapy, and the reason it works is it targets these rapidly dividing cells, so it kills the cancer cells at a quicker rate than it kills the normal cells. But I'm sure everyone in this room knows somebody who's been on chemotherapy and has seen the massive side effects to chemotherapy, whether it's the nausea and the sickness or the losing one's hair or the fatigue and the malaise. It's poison. There were no targeted treatments. What do I mean by that? Well, as we said, chemotherapy is very nonspecific. We just sort of give it and it kills cells. But targeted therapies, the idea is, well, what's unique about, say, one particular cancer? And that could be one type of cancer like breast cancer, or it could be within the context of breast cancer, one person's breast cancer versus another person's breast cancer, that could target the specificity of that. And if we can be more specific in what we target, we can ideally have treatments that are much less toxic and harsh. We also had no immune-based treatments. We're going to talk about sort of the immune system in cancer. This just wasn't recognized before 2010. There were a few people who were considered heretics out there who would talk about the immune system in cancer, but it was not en vogue and it was not proven, and that's just come full circle and been flipped on its head. And importantly, we had no pre-treatment predictors of response to treatment. The paradigm was basically, if everyone in this room had one type of cancer, we would look at you all as absolutely identical. We would put you all on one treatment, and if it worked, great. And if it didn't work, we'd change to a different treatment. But we couldn't pick one person and say, we have a test that says you're going to respond to treatment A, and a test over here, you're going to respond to treatment B. And so everybody was treated the same. And as you can imagine, that exposed a lot of people to a lot of toxicity that if we had a way to predict a better treatment, we would have. And that's not an indictment against what people were doing. It's just that's the best we had, even in 2010. And so, well, what happened around 2009 that sort of led to the current state of cancer in 2018? We had this, what I would call, and many people have called the cancer revolution. And the analogy I'll use is, well, what causes a revolution in something like this? And I'll give you an analogy to physics and the nuclear revolution of the early 1900s. And when Einstein came along, or before Einstein came along, there was sort of old Newtonian physics, and he came up with this radical idea of relativity. And that was just like an absolute new way of thinking in physics, and it led to all the developments of nuclear energy and nuclear power and the things that have sort of completely flipped physics on its head. Something like that essentially happened around 2000 that caused the cancer revolution. The first was something called the Human Genome Project, and we'll talk about that in a moment. And the second was the recognition of the role of the immune system in cancer. These two seminal events in the early 2000s really flipped cancer on its head. So we'll start and talk for a second about the Human Genome Project. This is a framed thing, a piece of, I call it art, on my wall. Science and Nature are our two biggest journals. It's like the New York Times and the Washington Post sort of level. And so to have an article in either one of these is a big deal. And when the Human Genome Project was published in February of 2001, it was published simultaneously by two methods in each of these journals. This was basically a 10-year, about $50 million effort to sequence the DNA of humans. And this was not done before because of technological limitations. And when it was done, it allowed us to look very deeply into what makes people people and what makes cancer tick. And it was just an absolute revolution and a new technology that now could be applied to lots of diseases, including cancer. The analogy I'll give you to this is in the literature. If we look at, people are familiar with Lewis Carroll and Alice in Wonderland. The sequel to Alice in Wonderland was Through the Looking Glass. And the description of this book after Alice in Wonderland is that Alice enters the fantastical world of Wonderland by climbing through a mirror into the world she can see beyond it. And this is a statue of an artist's interpretation of that sits in England. And I love the statue because it shows this person looking through this window. And this window for us was genetics and genomics and the Human Genome Project. We now had a way to look beyond individual cells and look down at the molecular level and see really what makes us tick. Well, why was this important? Well, let me use melanoma as an example. This is actually National Skin Cancer Awareness Month in May, so it's appropriate. Melanoma is sort of the poster child for success in this area. We as physicians like to look at these, what we call survival curves, where time is on the bottom and percent survival's on the top, and we don't like to see curves that look like this, where basically if everybody starts at time 0, but by a couple months we've lost a large portion of patients, and by 2 years almost everyone dies of this disease. And these were what we saw in melanoma because we just didn't have effective treatments. If you squint, you can make out the 2 lines on this curve, but they're kind of the same thing. And so that's what we were seeing in melanoma. And the problem was we could look through the microscope at a melanoma tumor and sort of say certain things about it, but we couldn't look beyond that. And so we were stuck in this era of the light microscope. And to give everybody some perspective, the era of the light microscope was long. The first microscope was invented in the 1600s. So this is the technology we had to call a cancer at that time. Well, then came along this Human Genome Project, and now we were able to look beyond the light microscope. That was sort of our window now with the Lewis Carroll analogy. We could really look into the cells more. And I don't expect anyone to have any interest or deep understanding of this picture, but this is a cell, and these are the different genes that make a cell tick with respect to skin cancer. And the important observation that was made because of genetics and genomics was that this one molecule, BRAF, B-R-A-F, had a mutation, a genetic change in it in more than half of melanomas. This was a huge deal. This was, oh my gosh, we found what makes melanoma tick. And importantly, it was the exact same mutation in all of these patients. So the same little substitution of one protein for another was causing these melanomas to form. And this was revolutionary. So a company started in a group of researchers, and this took a couple years, but they developed a little drug, a molecule. It's a pill form of a drug that you could take that would specifically target that mutation. And the important thing about that was this was the drug, the mutation, and it would target it with a high degree of specificity in the sense that it would affect that gene and that mutated gene, but not the normal version and not other cells. And so you could, like we were talking about, it was like a smart bomb, only going for that gene, sparing everything else. And so they started in mice, and they were able to get these tumors to shrink down. That's kind of shown down here. But importantly, very quickly thereafter, we're only 2 years later now, we have this publication in the New England Journal of Medicine, really our pinnacle of clinical medicine publications, that showed that targeting this, activated this BRAF mutation worked, and it worked remarkably. Many people may have seen PET scans or know what those are, kind of tumor scans. All of these dark black dots are tumors growing on this patient in their kind of upper arm area, and only 2 weeks later after taking this pill once a day, the tumors sort of melt away. And it was causing these incredible clinical responses. You'd have people who had performance status, they weren't able to use their arm or walk or move, and suddenly 2 weeks later are up walking and using their arm and pain-free. So it was a truly remarkable treatment, and it was one of the first to sort of have this kind of success. These are other ways that we sort of look at responses to treatment. We call them waterfall plots. Everybody sort of starts at 0, and if your tumor shrinks on treatment, you go below the line, and if it grows on treatment, you go above the line. And what you can see from this is almost everyone who goes on this therapy, their tumors shrink, some up to 100%, meaning the tumors go away completely. And that was never seen with regular chemotherapy. These kind of responses where everyone almost is below the line, never really seen. And we were seeing it in really hard to treat areas. It was quite remarkable. And this made this led to one of our a phase 3, a randomized trial, where to prove that it works, you sort of take patients and you say you either get treatment A or treatment B, and it's at the flip of a coin, and we sort of test that. And this worked remarkably well, and it made, this is a sort of from the New York Times again, multiple articles appearing in the New York Times and the lay press about the success of this. And it created a lot of controversy actually about the ethics of running a clinical trial when you have something that works that well. How could you ethically randomize someone to a treatment that works so well? It was sort of the analogy that we often say is, if you really need to do a randomized trial of whether a parachute works or not. No, you kind of just know it works. And that was really got into the ethics of this. These were 2 first cousins, both who had melanoma. One who got randomized to the sort of drug that didn't work, and one to this newer one, and a very interesting story from 2010. that gets into the ethics of that. It almost created a new field of oncology in cancer ethics. And you can see it took a survival curve that looked very poor like that to one that sort of looked a lot better, but still over the course of time came back down. So what we were realizing was much like we can put you on a treatment for a bacterial infection with an antibiotic, we can get resistance that develops, and we have to find something else that works. So we would go back to the laboratory, and they figured out that if you had this change in this BRAF, some of the cancer cells could sort of skirt around it and still replicate and grow uncontrollably. But what researchers then did is they developed another combination where they could give a drug that inhibits this gene and this gene at the same time. And so we came up with another regimen and now we're in 2015, where we give 2 drugs simultaneously, and we're able to take the curve that looked pretty good and make it look even better. And so by going back to the lab and understanding these treatment mechanisms of resistance, we're able to do that. And so now where we are in melanoma, everybody gets tested for this genetic mutation who has melanoma, because it allows us to give much more precise treatment. It's rapid and is inexpensive. And it's giving us understanding of how these tumors still continue to figure out how to grow, and leads us to develop even better therapies. Very similar to the antibiotic story. We're able to develop new antibiotics, combination antibiotics. This is combination targeted therapies. But they're pill forms, and they're specific, and their side effect profile is so much different and less than chemotherapy. So that was the kind of cancer genomics or genetics part of it. And well, what about the immune system? Because in what I would tell you is, if the genetics part was a great story, this is an even better story, even magnified by a 10 or 100, because it's really flipped our treatment thoughts for immune system. What happened around 2000 was some new laboratory discoveries in the cancer field that showed that cancer cells evade the immune system by basically co-opting our normal immune system, and it puts the brakes on the immune system. So our immune system has ways to turn on and ways to turn off, and you say, well, why does it need to turn off? Well, if you have a bad cold, and your immune system's revved up and you get a fever and arthralgias, you start to ache in your joints, that's because your immune system's releasing all these compounds to fight that virus. But if the virus was cleared and we couldn't turn that off, you'd feel crummy for a long time, and your body might start attacking your own systems, and you get autoimmune disease, and that's sort of a manifestation, autoimmune disease, things like lupus and rheumatoid arthritis of the immune system kind of gone wild. So a cancer cell sort of figures out how to stop the immune system from getting rid of that tumor cell. I told you that before that most tumor cells that form are cleared by your body's own immune system. Well, this gentleman, Jim Allison, who hasn't yet won but certainly will before his time comes, will win the Nobel Prize for really describing this. And what he described was, and I'll put the analogy to a brakes on a car, is the immune system has different ways to turn it on, a gas pedal, or in the case if you have an electric car, we call it an accelerator now, not a gas pedal, but or the brakes. So it has ways of doing either one of those. Cancer cells press the brakes, and they don't let off, because they don't want the immune system to recognize that cancer cell and get rid of it. So Jim Allison said, well, what if we can make the cancer cells take their foot off the brake? And in the laboratory, he realized that the interaction between a cancer cell and an immune cell has all these different proteins and molecules that govern whether the immune system is on or off. And he realized one of the most important ones that we'll sort of talk about, and developed a therapy against it. And when that therapy was given to patients, again with melanoma, and this is looking at about 2,000 patients, although most patients didn't respond to this one agent, what is different about this curve is that when it gets down to about 20%, it flattens out and it stays flat out to 120 months. So there were 20% of patients that were responding to this, and we don't use this word commonly, but were almost cured of their melanoma. What we were used to seeing was this curve go and steadily go down to 0, but this curve had this nice tail, and that really got people interested in the immune system's ability to treat cancer. This was with one drug and these really long-term success, and patients doing really well with it. Well, then, as we discovered more of these molecules and ways to inhibit them or treat them, we came up with combinations where we could give one drug, a better drug, or 2 drugs together, and now we're looking at patients, instead of at the 20% range, we're up to 50, 60, 70%, and again, over long periods of time, not just months, but years, and generally if somebody responds, they tend to respond for exceptionally long periods of time, if not infinitely. And so this immunotherapy revolution is here in melanoma. We have 2 FDA-approved drugs that we use to treat advanced melanoma in the immune system, and they work remarkably well. The long-term successes are completely unprecedented. It's flipped melanoma on its head. The toxicity of treatment, although is much lower, is different. These aren't sugar pills, so there are side effects, but much more tolerated than chemotherapy, much more specific. And now that we're developing even more and more of these combination therapies, that curve is raising up and up and up, almost in front of our eyes. So where are we going from here based on these this cancer revolution? Where is that taking us now? So we have even more immune modulating agents. And if I go back to that picture, I talked about sort of 2 of those molecules that have these FDA-approved drugs. There are for every one of these proteins or colored schemes across this spectrum, there is a clinical trial or active research being done to develop a drug that will target that. And the idea would be that you'd have all these different options where you could pull drug A and drug B together for a particular patient and a particular cancer type that would be very specific and predicted to work well. That's sort of the holy grail, and that's happening in certain cancers. In melanoma, lung cancer, kidney cancer are the 3 ones that are furthest along. Our challenge now is how do we take a disease like glioblastoma, brain cancer, or pancreas cancer, where if we give these single agents, they don't work. Try to figure out how to overcome that and make these therapies relevant across almost every cancer type. And again, the diagnostics to predict responses to therapy. So right now, if we look at, say, this drug combination of these 2 drugs, we get a nice response rate, but we don't have a way of predicting who's going to respond yet and who's not. And sort of we're still exposing patients to risk that hopefully we could mitigate by really understanding who's going to respond to what. And then what about vaccine and other strategies to augment responses? I bring this up for a number of reasons, but this was another publication of a cancer vaccine against melanoma that was published again in this journal called Science, kind of our pinnacle, our holy grail. Many people might recognize the name that's at the end of this, Jerry Lynette. Jerry was a medical oncologist here before he now runs the medical oncology group in Pennsylvania. Jerry's work to do this vaccine was funded by the Barnes Hospital Foundation. It's really expensive to do this, to take one patient, take their cancer, figure out what makes it tick, develop a customized vaccine. This isn't like the flu shot where everyone gets the same thing. This is this patient's vaccine. He showed that this is a viable strategy, got a huge grant from the NIH to really push it to the next level, and this is something that we're all unbelievably excited about because this would be the most specific therapy. We sort of get your immune system ramped up and give it a boost with a custom vaccine. This wouldn't have happened without the Barnes Foundation, I guarantee that. And so I sort of crossed out melanoma here because I want everybody to understand this is now going to be, this is really sort of my thoughts on applicability across all cancer types. Melanoma has sort of gotten us in a lot of ways to where we are with immune therapy because it's sort of the poster child, but we've got a lot of progress where we can make this even more personalized and precision so that if anybody had cancer in this room, you would be treated as a very individual. We would take your tumor, we would sequence it at the genetic level, we would test it to see what are the best therapies that are going to benefit you, that are going to be least toxic and most effective. We're getting much better diagnostics where we can sort of both prognose and predict who's going to respond to what, how someone's going to do, so we can sort of select what's the best risk-benefit ratio of a particular treatment. We have much better improved imaging. We're so fortunate to be at a place at WashU and Siteman where we have the Mallinckrodt Institute of Radiology where some of the best imaging research is being done and pushing the envelope, and I'll allude to that in a moment. But a lot of, I think the foundation has supported a lot of work at Mallinckrodt as well. And extending the success of these therapies to other cancers is the goal and the key in the area of a lot of active research right now. And this has really tilted the balance of efficacy with morbidity. So we have much more specific treatments that are by definition much less toxic. People are able to go around and not only have a longer life, which is important, but a quality life. They're able to do the things that they want to do while they're on treatments. It's obviously no good to the patient or the physician or really anyone in general if we keep you alive but with a poor quality of life, and that's not good for anyone. This the cancer revolution I've talked about has really flipped that. So what I'm going to sort of show you is some advances in imaging. This is on the top are sort of a regular CAT scan, and with these newer imaging techniques, this is a patient with a tumor of the pancreas. You can see it lights up like a Christmas tree right there, and that's because this is work done at Mallinckrodt where they've developed a specific molecule that they give into you right before you get your scan. If anybody's had a CAT scan or a PET scan, you know you get the contrast beforehand. This is a tumor-specific contrast now, so the only thing that's going to light up are certain things within the tumors, and what this has allowed us to do is not only see the tumor, but if the patient has spread of the tumor outside of the tumor, we can see little spots where the tumor's spread. Well, why is that important? Well, I do a lot of pancreas surgery, and if we're going to put someone through a big pancreas operation, that's a big surgery, and there's a lot of side effects, and we wouldn't want to put someone through it if the cancer had spread outside of the pancreas. So having better imaging like this also allows us to much more precisely select the best treatment for a patient and spare them an unnecessary surgery or surgery that's not going to benefit them. So huge advances in imaging. And this has been then studied also in the New England Journal of Medicine, and not only can we use it for imaging, but we can take that tumor-specific agent and put a therapeutic, and we can merge those two molecules. So this was a study where we took that imaging agent, hooked it up to a small radioactive particle, but instead of that radioactive particle going to the whole body, it only goes to the tumor. So again, very precise kind of smart bomb type approach for these types of tumors. This is being extended to a lot of other tumor types. This is a figure from some work that's in my lab where we're really trying to understand cancer evolution, again, how this primary tumor turns into a metastasis or spreads. We got a grant that was funded through the Barnes Foundation and the Cancer Frontier Fund and Pedal the Cause. I'm wearing my Pedal the Cause Foundation tie today for that. But this was nearly $1 million grant that we got that allowed us to study patients with colon cancer where it had spread, and we could get all these different tumor specimens that were throughout these patients' bodies and really study them at a very deep genetic level. And what we were able to show was, what I want to point out is all the different colors here. So it wasn't just one color, one cancer cell, and they were all the same, much like people in this room again are different. The cancer cells within individual patients' tumors were very different. And when they spread to the liver, certain cells that were in the primary tumor were the only ones that showed up in the liver. And so we're now being able to understand at the genetic level what allows a cancer cell to spread to the liver, and we're hoping that leads to even newer therapeutics. And what we've really been able to do that's exciting, if people in the investment world in the audience, we've taken that $1 million investment from the Cancer Frontier Fund and the foundation and turned it into about $10 million of NIH grant money to sort of push this forward the next level. If we would have tried it the other way around, just sent this idea to the NIH or the government, it wouldn't have worked. They would have said, well, where's all the data you need to sort of say that you can do this? And so that's where the foundation is so critically important. So what I would say now is we're sort of pushing through the looking glass. We're kind of getting through this cancer revolution and the genetics that we have now, and there's going to be another looking glass down that's the next one. How can we look even deeper past the genetic level? And people are looking, are doing incredible research that it's sort of pushing the envelope even further. What are the challenges that I sort of see now in the future successes? Well, one is things are unbelievably complex now. Where we used to have breast cancer and melanoma, now we've got breast cancer divided into 10 types at the genetic level. And the massive amount of information that you get from doing these genetic sequencing experiments are terabytes and terabytes of data for every patient. And then being able to share that with other investigators and compare, it's created a whole new world of cancer informatics. People with PhDs in mathematics and biostatistics and things that are the ones that we need to study this and be able to make sense of it because they're such large data sets. The medical system structure is very challenging because of the way that we sort of have the past paradigms of sort of if you have a particular cancer site, we treat you with this regimen and then we do this and that. There's not sort of this tumor or cancer type agnostic approach where we don't care if you have breast cancer or colon cancer. If the genetics look like this, we want to use this therapy. And that's a completely new paradigm shift that has implications at all the way down from the hospital level to the physician level. So the structure has to change for this to work. And of course, cost. And this cost I can put at multiple levels, whether we're talking about clinical care and the cost of these drugs, and you see it in the lay press all the time, what they're charged and what it costs, what it takes to develop one of these drugs. The loose sort of term that or amount that I'll give everyone is if I had a great idea today for a cancer drug, it would take about 10 years and about $1 billion to get that to market through all the clinical trials and studies and tests that you need to prove that it's safe and effective. And we're very conservative, much more so than, say, Europe or Asia, not in a bad way, but in a way that safety comes first. But that's the reality of what it takes in terms of cost and time. It's also the cost of the research. I mean, we've talked about it a little bit, but the NIH budget has sort of stayed flat for a while. It's picked up a little bit, but it's still very hard to get research ideas funded. Whether it's a good idea or not, you still need a lot of preliminary data to sort of support your idea, and that costs money, and that's where things like the foundation are just absolutely invaluable. And whether that's a basic research study or a clinical research study, it all comes out the same. And this is true not just for cancer, but I think all of biomedical research. So I'm going to end there. I think we'll have a little time for some questions, and I'd be happy to talk with anyone offline as well. My contact information is here. Obviously, we'll be here for a little while, and have really enjoyed the opportunity to interact with the foundation over the years. It's just a fantastic organization and a group, and I can tell you that we, as physicians in the cancer field and my colleagues in general, we don't take it for granted, and we're just unbelievably grateful for your support. Thank you. I saw a hand. Yep. It's almost a perfect analogy to any technology, and the one that sort of started out was the computing field. So it halves, it comes down by half about every 2 years. And so it's remarkable how quickly it comes down as the technology efficiency improves. The Human Genome Project was literally a $50 million project, and that was around 2000. And we've come to the point where if I drew a blood sample from you and we wanted to sequence your genome, it would cost about $1,000. The part that is a little bit more hard is then, okay, so we've got that data, and how do we analyze it if we were to do that for everyone in the room and wanted to do some comparison? And that's where it's person expenses that come in. You need full-time people with bioinformatics background to do the analysis, especially when you're doing it at the research level. So we can't just take a computer and say, okay, tell us who's at high risk for cancer A and B. We're sort of diving deeper into it where there's no computer that's going to do it. It's still a lot of person power. Yeah. Dr. Cotter.
That's a terrific presentation. In my mind, the foundation is a bit of the face of the medical school to the community. So it seems that right after the community gets a McDonald's, they have a cancer center. How institution specific are these exciting new advances?
Mm-hmm.
Does the person, our friends and family, do they really need to come to us, or are they going to be okay in a small community?
Yeah, no, I think that's a great question, and it's one that's directly relevant, say, in a catchment area like like Wash U, because we don't have another cancer center, NCI cancer center right across the street or 20 miles from here. And so we get a lot of patients from rural Missouri and southern Illinois and all over, and the question is, do I have to get my treatment here or should I? And for some of these newer therapies, it's pretty important because the experience with what are the side effects and how to best select patients for these are critically important, and although it's spreading out to the community, it will take time. And then I think the more important thing are the clinical trials and the ability to participate in a clinical trial that's testing what we have now versus something we might think is better. And so that's, I think, one of the keys and one of the most important things that we draw patients in for. And when we, and I say we meaning Siteman Cancer Center, was evaluated by the NCI and the NIH at our last cycle, we had a perfect score in that area, and that doesn't happen very often, and that area being our ability to bring patients in from afar and treat them on clinical protocols and do that in a safe and effective manner. So critically important. Yes?
I'm interested in the patients who are not being given the experimental drug.
Uh-huh.
And if there is a success, do they then get it?
Yeah, so what's evolved from this sort of ethical dilemma and the way we sort of think about these clinical trials is if somebody's being treated for say cancer A and they're getting a drug versus a true placebo, they're getting nothing, and they progress, then usually there's what we call crossover. They're allowed to sort of then get the drug or get the experimental drug. The other thing that a lot of these trials have now is, let's say the trial was supposed to go on for 2 years, but after a year they do an interim analysis. They look early and they say, this is so positive, it's so overwhelmingly effective, we're going to stop the trial and then and make this an effective therapy. And then usually in short order, the FDA will approve it. And once the FDA approves it, then it's not only available for me to prescribe for that indication, but almost all payers then pay for it. Yes? Yeah. Yeah, that's a great question, and it's one of the initiatives of the National Cancer Institute for this whole field of research money that's set aside for early detection. And so there's a few cancer types where there's sort of a paradigm for that. There's one example I'll use is there's a particular genetic mutation that if you have it, you're very predisposed to gastric cancer, stomach cancer, to the point where we recommend removing one's entire stomach so that you don't get that cancer, because it's almost 100% that during your lifetime you will get it. The other area of research where this has sort of changed the field is thyroid cancer. I mentioned Dr. Wells, who was the chairman here. He discovered the gene that caused this hereditary thyroid cancer, that kids would essentially have that cancer when they were born. So the way that is now treated is these children, when they're several months of age, and the reason you wait several months is the thyroid gland is so small at birth, you just can't do it right at birth. But you operate on these children at several months to remove most of the thyroid, and almost all of them have thyroid cancer even at that time. That clusters in families, so oftentimes we know the families beforehand and they have the genetic testing, but that's completely changed the way we think about it. What we would love to have is in an area like pancreas cancer, where 90% of the time when it forms, it's not curable. We'd love to have a way to say, hey, we know you're at high risk for pancreas cancer, so we have this screening test that we're going to pick it up in the blood before it's detectable. There's ways now that are sort of becoming very in vogue of being able to test one's stool for colon cancer and one's bladder for kidney cancer, for bladder cancer, that are sort of becoming non-invasive ways to monitor things, especially for high-risk individuals. Sir?
Do you collaborate with any other research institutions?
Yeah. Great question. It really gets to the way that the big data sort of side of things gets. Is it would be silly for us to be doing all this genetic sequencing and we're looking at it, but the rest of the world can't look at it. And so one of the big shifts in the way the NIH funds research now is they want collaboration, and they want you to show a way that you're not just sharing your data, but actively collaborating on it. So we have a couple large-scale research grants in the sort of several $million range where there's 5 or 6 centers all working together. Usually there's one data coordinating center, but the grants are written, and they're, the only way you get it is if you're working with a number of centers and catering to the strengths of different institutions, bringing all of that data together with hypothesis-driven research, and it's sort of a very synergistic and symbiotic way to move the needle. Yeah. Yes. Mm-hmm. Yep. No. Yeah, correct. Not generally, definitely not the case where it's one pharmaceutical company. Yeah. Yes. Mhm. Usually, so glioblastoma, which is kind of what most people think of brain cancer, like Senator Kennedy, who died of this disease, that's the typical brain cancer is glioblastoma. And it typically doesn't spread, but the problem is it grows in the brain, and we're limited on what you can do surgically in the brain. And if it grows and expands in the brain, of course, it causes pressure, and pressure causes problems. So it's one of the most, if not the most, lethal cancers. The incredible amount of research that's going into glioblastoma and making strides is being led by places like Duke and WashU, where here we have Gavin Dunn and Albert Kim, two neurosurgeons that are very well funded, have been funded by the foundation and Peddle the Cause, who are doing very interesting immune system research, and some of the work that was started at Duke, which is involving a therapy that has taken a polio virus and modified it so it attacks glioblastoma cells. And that people may have seen on the New York Times or Charlie Rose and these sorts of things. It was a big deal when it came out about a year ago, and is really kind of getting to the point where it may be ready for prime time soon and may really, really crack what's been a very tough nut in glioblastoma and brain cancer. It's not hereditary, correct. Correct. Yep. Well, again, thank you all. I'll be here for a little while, and feel free to contact me. Thank you.
I tell you, every time I listen to some of these research physicians from WashU and Barnes, I think how lucky we are to live in this town near all this research, and how advanced we are becoming in the last 6 to 8 years. Excuse me. And it's a big tribute to their ability to do the research that they're doing. And as Ryan told you, a lot of this is started by foundation grants and Peddle the Cause funds and so forth. So your money and your gifts and your legacies are well put and in very good hands with people that are making things happen that will affect the health care of all of us in our community and our friends. So it's very heartening. And I'd also say that was a great presentation. I mean, you took it down to the right level where I think we could all understand it and answered our questions. And we've got others that the foundation sponsors. We have leading edge presentations that happen. We got one coming up in July and another coming up in September. And we'll let you know when those are coming, or you can look on our website. And they are terrific. And we get some of the best physicians and best researchers in various areas, and they come in and talk about advances that they're making, and just as Dr. Field said, what the future looks like. And so I would highly recommend those to you, and we'd be happy to get you the information on all of them. So again, Dr. Fields, thank you very much for a very, very good presentation. A lot of this is because of all the people in this room and the vital role that you play in helping us and supporting us, and it really is resulting in some great outcomes. So thank you for joining us. Thank you for giving us your legacy, gifts, and continuing to be involved in the foundation. And if you ever have any questions about the foundation whatsoever, just give us a call. We'd love to answer them. So thank you for coming, and have a wonderful afternoon.